So I'm Joan Steitz. I've been a professor at Yale since 1970, late 1970. And just want to add that ever since I started in science, my passion has been RNA molecules, their structure and their function.
I'm Christine Guthrie. I've been a professor at UCSF since 1973, although I'm now retired. I first met Joan in 1969 at a meeting at Cold Spring Harbor on RNA.
I'm John Abelson. I was a professor in the chemistry department at UCSD from 1968 until 1981, and then I moved to Caltech, Division of Biology, and I was there from 1981 until 2004, and then I came here and I worked in Chris's lab for 10 years. Joan and I have known each other even longer. We were together in Cambridge in the 1960s, 1966 or so. And so it's a long relationship between the three of us.
So Joan, maybe you could share with us what some of your early influences that caused you to go into science.
Well, I was very fortunate, as I think is true of many, perhaps not all, women scientists, to have a very supportive father. He was a high school guidance counselor, but I think he really had wanted to be an engineer and somehow felt he wasn't up to being an engineer, so he was very encouraging of my interest in science.
Very nice.
And that's basically what got me to being a chemistry major in college.
Nice.
What about you?
My mother was a writer and my father was an artist. And my mother said, "Do not be an artist." And my father said, "Do not be a writer."
And
It
what
was
was
sort
left?
of default.
And John, in high school, what was your interest?
I was interested in football and basketball and track, and I would not have gone into Caltech. There's no way I'd ever gone into Caltech. And when I got to Washington State, it turns out that I got a very good education, and I got turned on by science. Joan, I'd like to ask you, we were both students in undergraduates in the 1950s. And although the DNA structure had been determined in 1953, there was nobody where I went to Washington State that had the slightest idea about DNA structure. But what about you? You were at Antioch. Did you learn anything about molecular biology there?
Well, not in my courses, because it was just too new. So I arrived at Antioch in 1958. I very shortly thereafter became a chemistry major, but DNA was still so new, having been described in '53, that it hadn't made it to textbooks or even to courses as yet. And I was fortunate in that I was at Antioch that had a work-study program, and it turned out very fortuitously that there was a job that was open for chemistry majors and people like that in Alex Rich's lab at MIT. And Alex Rich was one of the sort of founders of the field of molecular biology. And I remember when I arrived in the lab and learned about the structure of DNA, I was just completely enthralled, because as a high school student, I'd been sort of fascinated by genetics and what the molecular basis of heredity might be. And here all of a sudden was this beautiful molecule that could replicate itself into two pieces, each of which had instructions for the other. And this seemed to me to be the way that cells might actually operate to pass on genetic information. And I remember just falling in love and knowing that this is something I really wanted to do more of. So I was very fortunate in getting that position.
Who were the people that you worked with in Rich's lab at MIT?
So I worked with primarily Jonathan Warner, who subsequently he spent most of his career at Einstein. He was a graduate student, actually, and he's done many, many very important things on the ribosome. And Paul Knopf, who was a postdoc who subsequently made a name for himself in the field of immunology. And with John, I was actually doing quite an incredible project. At that time, it was known that you could take the DNA molecule and heat it up, and it would come apart into its two strands, and the optical density would go up because you were going from double strand to single strand. But then you could lower the temperature back down and get the DNA molecules to reform as duplexes. And somebody had the bright idea that maybe the same thing was true of ribosomes. Even though it was known that they had RNA in them, nobody had any idea of whether it might be double-stranded RNA or not. So I spent most of my time standing in front of a spectrophotometer, raising the temperature,
the Carey spectrophotometer, right?
DU spectrophotometer, and slowly lowering the temperature in all sorts of different buffers. And of course, we never saw the OD go back down because, of course, we now know that the proteins came off and denatured, and ribonucleases chewed up the RNA, so there was no way that one was going to get back a ribosome.
It was
by
an
that
experiment before its time.
But nonetheless,
yes.
I liked it a lot.
Yeah.
So I had several jobs as an undergraduate, but I decided that I would go to medical school rather than try to be a research scientist, because when I looked around, I'd never seen a woman professor or a woman head of lab. And it just didn't enter my mind that this was something that was possible for me. So I applied to medical school, and I was about to go off to Harvard Medical School when the summer before I wanted to be home with my parents, and I found a summer job that happened to be in the lab of Joe Gall, and you guys know who Joe Gall is. He's just a really, really fabulous cell biologist who did things like, invent in situ hybridization and discovered the octamer structure of the nuclear pore. He was an assistant professor at the University of Minnesota. But that summer, he was in the process of moving to Yale, which was where he was for then several decades after that. And so he just sort of gave me my own project and was packing his boxes and mostly wasn't there. But it was the first time I'd had my own project. Previously, I'd worked for graduate students or postdocs and the other lab experiences I'd had. And for the first time, by August 1st, I said, maybe I really don't want to go to medical school. Maybe I ought to go to graduate school and be able to continue doing this, regardless of what it meant about my future prospects. So I talked to Joe Gall about it. I had met Jim Watson, who was a sort of competitor friend of Alex Rich, when I'd been earlier at MIT, and somehow it happened that I was able to switch from the medical school into a new graduate program in biochemistry and molecular biology.
Watson is a figure of renown.
Yeah.
And very briefly, from my perspective as a graduate student and reading the Journal of Molecular Biology, the Watson lab was a fabulous place.
The Watson lab was a fabulous place. It was so exciting, and there were so many people. There were people working on ribosomes and people working on the mechanism of protein synthesis and people working on the RNA phage, R17, that I ended up working on. And it was a time in molecular biology when there were maybe 50 labs in the entire world that were doing it, and they were all in communication. And we sent around preprints. I don't know if you know about this. There was a group called the IEG, the Information Exchange Group. Everybody, sent their preprints around so everybody else would know faster. what they discovered, and Jim would go off and talk to people like at Rockefeller, our great competitors at Rockefeller on the RNA phages, and he'd come back saying, oh, they've done this, they've done this, they've done that, and everybody would get all excited, and it would be another spur
So it's
to move
interesting
forward.
that as early as that, the field was uniquely cooperative
Yes,
rather than
yes,
competitive,
Yes, yes.
which has, of course, since gone on
Yeah.
to characterize the whole RNA field.
Field, right. So this was a field that was interested in what the basic mechanisms of living cells were. And it was, of course, fervently believed that if you didn't start simple by working on bacterial cells and their viruses, the bacteriophages, everything else was so complicated that you would never learn anything. So you had to stick to these simple systems. Even to the point that I remember people who were trying to work on mammalian cells being derided because it was so complicated that they would never figure out anything, which we now know, of course, is completely untrue, but that was the attitude at that time.
And it was likely at that time that they didn't learn too much either.
Well, at that time, absolutely. But, I remember, I just couldn't conceive of knowing the 4 billion base pairs of the human genome, or of
anything,
a
of
biotech
anything, actually.
industry that's worth now trillions of dollars, right? And what we know about DNA, saying so much about evolution and being useful in medicine, and it was just beyond belief, inconceivable. Now I know Jim, who always had good sort of future insights, he could see these things, and that was partly why he became head of the Human Genome Project when it was just being initiated, because he could see that these things were going to happen. But boy, it was way beyond me. It was just a matter of trying to figure out about bacteria and bacteriophages.
It's all in the timing.
Yeah.
It is in the timing, and I would say for the record here that the three of us have been blessed by when we were born.
Yeah,
Because
exactly.
there couldn't have been a better time to have been born and ended up doing biology.
Right.
Because we went from this period in which we didn't know anything, basically, and the things that we did learn were quite beautiful and led one to think that life would be simple. And yet, now we know a lot more. We know that life is not so simple, but nevertheless, we do know many, many basic mechanisms. And so it's been an incredible journey of these 60 plus years.
But since we want to talk more about sort of the beginnings of understanding splicing and so on, I think it's important for both of you to say a little bit more about, how you got to the places that we all met.
Well, for me, it was, I had come from a history, as Joan said, working with simpler systems, with bacteria and their viruses. And I had focused on the RNAs, which were really the only ones for which there was any structural information, which were the transfer RNAs. And I took a basically genetic approach to looking at that, and we isolated things we called suppressor tRNAs, and then could analyze their function. John and I actually started out as competitors. We were probably the only two people in the world interested in these bacteriophage tRNAs.
But you'd done genetics earlier as a graduate student,
As a graduate
right?
student, I had done genetics actually of ribosomes and ribosome synthesis. In those days, originally it wasn't understood how you synthesized an RNA, and part of this early work in my thesis established that there were mutants that accumulated intermediates in the synthesis of ribosomal RNA. And so that was in a sense, the beginning of RNA processing, although we didn't call
And I
it
remember,
that.
I think there was a big discussion about, say, chloramphenicol, which clearly also interfered, but it wasn't clear whether what accumulated was in fact an intermediate or some sort of secondary product that was a dead-end product in ribosome biogenesis.
That's right, and it was controversial for some
Yeah,
time.
yeah.
Yeah. Anyway, so that got me interested in the biosynthesis of RNAs in general, although the question of messenger RNA biosynthesis turned out to be much more complicated once people started looking at mammalian systems.
So let's have John go back and say a little bit about how you got to the MRC labs
I'd
where
like
we
to
met.
start a little earlier. I was a student of physics at Washington State University in Pullman. The head of the department there, Paul Anderson, remarkable man who actually had gone to Cambridge in the 50s and seen the structure of DNA. Crick had shown it to him, and he talked about it. And this was really exciting to me. And so I decided to go into biophysics instead. And I looked around the various programs, and I ended up at Johns Hopkins in the Department of Biophysics. And there was only really one person that was doing molecular biology at Johns Hopkins. His name was Charlie Thomas. He had been in Paul Doty's lab at Harvard, and we did physical chemistry of DNA molecules. And so there was, I didn't do anything with RNA as a graduate student, but we were very excited about what was going on in our department, the students in particular. And for example, the genetic code was really being worked out right next door by Marsh von Nuremberg at the NIH, and we had joint seminars with them and so forth. So the excitement of molecular biology was very much there, although I wasn't doing anything with RNA. So it was only when I came to Cambridge that I began to work on RNA.
So the way I got to Cambridge as a postdoc was because it was a mecca for X-ray crystallographers. And I had married my husband Tom, and he was an X-ray crystallographer, so we had to go to Cambridge. And when I arrived in Cambridge, I recall Francis Crick, who I was theoretically working for because Jim had arranged it with Francis that there would be some space for me in the lab. Francis said, well, there isn't really any space for you. Why don't you do a project in the library? Now, I immediately knew that theoretical whatever was not my forte compared to experiments. And so I went around and I talked to all the people in the whole division. This was a division run by Francis Crick and Sidney Brenner, whose death we're mourning this week, and tried to find out what they were doing and whether there were good areas that they thought, an additional person could work in. And one project that was suggested to me by several people was one that clearly my male counterparts, the other postdocs, had considered but rejected as being too challenging, was the idea of binding ribosomes to a messenger RNA, trying to get out the pieces that were protected, and then sequencing those using the new methods that Fred Sanger upstairs was just developing for RNA sequencing that had been so potent with respect to tRNA. And since I had no vision that I would ever need to go back to the States and apply for a faculty position, as all the men did, I said, gee, this sounds like an interesting project. Why don't I take this on? And Mark Brecher, who was a staff member at the MRC, gave me sort of this much space in his lab, or on his bench, I should say, because I think the lab wasn't that much bigger than his bench, and said, go ahead. And he was a wonderful counselor and very, very smart scientist, and really helped a
Incidentally,
lot.
none of us had any more space than that.
Right. So that was how I got into the project of actually looking at messenger RNA and trying to figure out about translation and where protein synthesis begins when ribosomes translate, as they say, polycistronic messenger RNA, one that has multiple genes or open reading frames on it.
But you brought to the table the small RNA phages.
Right. So I had worked on the RNA phage in Jim Watson's lab, primarily on one of the three major proteins that's encoded that is part of the structural capsid of the viral protein, and that's responsible for it being attached to its host during infection. So I knew how to grow these phages, and I knew how to label them so we could get very highly radioactive P32 labeled RNA out of them, which was a prerequisite for this other project. And as you know, John, for about a year things weren't working very well because I was basically using the wrong procedure for isolating these ribosome protected fragments. And we had a lot of fun going to antique auctions and doing other things when experiments weren't going well. And then all of a sudden, I changed part of the methodology. I remember Sidney saying to me, he said, experiments are sometimes like a bad marriage. You have to give them one more try before you give them up. And I was on the verge of giving up, and so I tried in a slightly different way, and all of a sudden it worked, and I was able to get these fragments and sequence them.
Because it was exciting that you actually pulled it off.
Technology is always important. I've always appreciated.
How did you feel about the amount of P32 that you needed to use in these experiments?
Well, I think we didn't have a scale, or at least I didn't have a scale in my head. Later, it became apparent that the amount of P32 we were using was pretty extraordinary. Nobody nowadays would ever do experiments, a single person with that amount of P32, but everybody was doing it then.
And we used to mouth pipette it.
Yep, you had to be very careful not to get it into your mouth.
Yeah, I think it's worthwhile saying that what was really so fabulous about being in Cambridge at this time was that, on the one hand, on our floor, the second floor, there was genetics going on. Sidney Brenner was a geneticist, and his scope was very, very wide. He's really, I felt,
A real
perhaps
genius, absolutely.
A genius, perhaps on the scale of Mozart.
Yeah, I agree.
Nice analogy.
Yeah.
So in any case, I learned how to do some genetics. Upstairs, there's the fantastically different kind of person, Sanger, very, not like Sidney at all, but perhaps having learned first that the proteins actually have a sequence. Then he learned how to do sequences of RNA. So we were doing the very first sequences of RNA there.
Yeah. And his methodology was brilliant.
So we were very, very far ahead of the rest of the world.
Absolutely, yeah.
Yeah.
It was very exciting to see those tRNA sequences and then the fragments of messenger sequence and so on. They were the very first insights into the fact that you could confirm, for instance, the genetic code by knowing the amino acid sequence of the protein product and comparing that and seeing that the genetic code that had been deduced was correct.
Yeah.
So my husband and I both went as assistant professors to Yale. Meanwhile, John, you'd gone to UCSD.
Yes.
And Christine was still, what were you doing in 1970? Were you still a graduate student?
I was just finishing my PhD and starting a postdoc in Berlin.
On tRNA or on ribosomes?
Ribosomes.
Ribosomes were a big thing in Berlin, with the laboratory of Wittmann.
Absolutely.
And Chris
He was the king of ribosomes.
Yes. But they all thought that the proteins were the thing.
Exactly. And the RNA was just a rack on which
Yeah.
you hung the protein.
Which went on for some time.
Which went on for some time. And I like to think that there wasn't much that Fred Sanger got wrong, but one sequence that had been done was the 3' end of the 16S RNA in the small ribosomal subunit. And Shine and Delgarno resequenced it and got a different sequence, and then realized that in the ribosome binding site sequences upstream of the initiator AUG were these sequences that appeared to be able to base pair to the messenger RNA. And I got very excited the minute I heard that idea and spent a lot of time trying to figure out how to prove it, and eventually came up with... with the first biochemical evidence, but thereafter followed genetic evidence, and that's what we want to move forward with talking about splicing.
You've always had very good judgment about the importance of interactions, and your judgment was correct on that. But at the time I thought, geez, I mean, we only know a small amount of the sequence of ribosomal RNA, and we only know a small amount of the sequence.
How can it
of
possibly be
RNA.
correct?
How could it be?
Right, yeah. But it turned out to be, turned out to be correct.
When you get the crystal structure finally, that's what you see.
Yeah. So you guys were working on tRNAs and suppressor mutants of tRNAs, and I was working on the initiation of protein synthesis, which I did for more than the first 10 years that I was on the Yale faculty, and a little bit on RNA processing.
When did you start with SNRPs?
Okay, so the way that happened was in 1976-77, Tom and I had a sabbatical coming up. And in those days, one actually, went away and you weren't really in touch with your lab because the only thing you could do was write letters or do telephone calls, which were extremely expensive. So I was away for a whole year.
The facts hadn't even yet appeared.
Of course, faxes hadn't yet appeared. And the early 1970s, I feel, I don't know how you feel, were a very interesting era in molecular biology, because by that time we knew the basics of DNA replication, some of the basics of RNA synthesis, quite a bit about protein synthesis and the factors, but all in bacteria. And some people were starting to work on higher cells. And a lot of people felt that, well, the golden days of molecular biology are over. We know all the basic principles.
Gunther Stent.
Exactly. Wrote about this molecular biology being dead. Right. Because everybody thought when we went to higher cells, there'd just be more bells and whistles, but nothing fundamentally new, which we now know was so wrong.
Well, it's very interesting. The genetic code was it did seem that was the real thing that you needed to know in order to understand life. And it is beautiful. There is just one genetic code. That leads you to think that life is something like physics. That is, and once you know these things, then where are you going to go from there? That's
what it was like in the early 1970s. But there were some things that were sort of didn't fit the picture. And being in the RNA field, I was aware of these. Okay, one was the too much DNA phenomenon that you all know about, that a phage genome with 100 genes has about 1/10 the amount of DNA as a bacterial genome with several thousand genes. That made sense, but then when you went to mammalian cells, there was 1,000 times more DNA, and nobody thought that we would have 1,000 times more genes than bacteria. So there was too much DNA, and then there was the RNA problem. What had become clear was that there was lots of RNA made in the nucleus of particularly mammalian cells, but only a tiny fraction, like 10% of it, ever made its way to the cytoplasm in the form of messenger RNA. And there was all this turnover and degradation in the nucleus. And so when we had the sabbatical, I thought, well, maybe I should think about something adventuresome in higher cells. And what was known was that there were conserved abundant RNA binding proteins, what we now know as HNRNP proteins, that bind to the nascent RNA once it's made in a eukaryotic cell nucleus. And I thought, well, maybe these proteins have something to do with deciding about which bits of the RNA go out as message and which stay behind to get degraded in the nucleus. And I wanted to study this, and I tried to make antibodies against these proteins. And even though I was in an expert lab of making antibodies, Klaus Weber and Mary Osborne, I never got antibodies because these proteins are very highly conserved and very non-immunogenic. So I gave up on that project after 7 months and did something else for the rest of my sabbatical. But that was the year, the summer, when splicing was deduced.
But
I
there
would
had
say
to be
by
splicing.
by the entire molecular biology community. I mean, there were people all over the world who made observations that they just couldn't figure out. And then all of a sudden it came together. I was not there, but I know it came together at the Cold Spring Harbor meeting in June of 1977. And then shortly after, we had the papers from Rich Roberts' lab and Phil Sharp's lab that said, okay, in adenovirus we can actually see these amazing structures, and we can see that part of the RNA is cut out and discarded and the rest is spliced together.
And for me, much more convincing was the deduction of Philip Leder's lab showing that hemoglobin gene has two introns.
Two interruptions, right.
There had to be new words to do.
Yeah, right. Yeah. And there was the time when they were called intervening sequences versus introns, and introns sort of won in the end. Yeah. So, but were you immediately interested in that?
Absolutely. I was interested in this, but the root to it was that both Chris and I had decided by this time that we should move to a eukaryotic system. And I took the yeast.
Because it was the early days of cloning.
Yes.
And that was starting to work.
Transformation.
Right.
Yes. Transformation especially was a big one.
But Joan, you've left out the part about how you went from
Ps
HNRNPs
to SNRPs. Right, okay. So when I got back to the lab in the fall of 1977, everybody wanted to work on splicing, but we were totally clueless about how to proceed. And then in January of 1978, there appeared a new issue of Nature, and at the back of that issue was a short article, one of the reports, about that mentioned something about autoantibodies. And the reason it caught my eye was, well, I'd been trying to make antibodies the year before and failing. Several people had said to me, I think I've heard of patients that make antibodies against something in the nucleus that has RNA and protein in it, which is what I was trying to get. And maybe you should be, looking at that. And at that time, I hadn't known who to ask. But when I got back to Yale, I had in my lab Michael Lerner, who was a brand new MD PhD student. And when that article came out and mentioned these autoantibodies, I said to Michael, do you know of anybody here that might have these patients? And Michael said, sure, I'll go see Hardin. Now, Hardin was John Hardin. He was the head of the rheumatology section of the Department of Internal Medicine. His lab was directly across the street from ours. And Michael went over there and came back that very afternoon with various little vials of patient sera and started to work with them. And what I'm always reminded to say is that, of course, if that occurred nowadays, one would have to spend weeks, if not months, maybe years, filling out human investigation forms, and we never would have done it. But there weren't those regulations in 1978, so he came back and immediately started trying to work with them. And then there were various other hiccups that happened after that. But eventually it turned out that the autoantibodies in some of these sera were directed against the SNRPs, the RNA protein complexes involved in splicing.
Which you found everywhere except in yeast.
Absolutely, yes. I wanted to discuss that. Okay, so Christine, how did you get into this then?
I just never bought it that it would be true of every other eukaryotic cell and not be true for yeast. I just fundamentally rejected that.
Mm-hmm.
And although people thought I was crazy, and I think in one of your papers you actually published that you didn't see them in yeast.
In yeast. We hadn't looked very hard.
Well, in retrospect,
Yeah.
it was a question of
yes.
abundance. But most people thought I was crazy to insist that they would exist in yeast. And of course, yeast was kind of lagging behind in the whole intron issue. Wasn't clear how many introns yeast had. It was thought that even if it had them, they would be very different, in part because they had a conserved sequence within yeast that no one had seen in mammals, the so-called tac-tac box.
Yeah. And one of your postdocs actually isolated something that we thought would be one of the snRNAs, and it turned out to be a nucleolar
That's right.
RNA, right?
That's right. I mean, finally, the RNAs we looked at, we identified by getting antibodies to the unique cap structure on snRNAs. Reinhard Lerman had isolated antibodies to the TMG
Did
cap.
a lot of beautiful
And
things.
so when we did that precipitation, suddenly they went from invisible.
Yeah. But I
to
also
visible.
remember when Tom and I were in your lab in 83, 84. So this is like 4 years later for a sabbatical. At that point, again, Reinhard Lerman had been part of the discovery that if you used complementary oligonucleotide in RNase H, that you could then inactivate the U1 SNRP. And I was confident that if we did that experiment on a yeast extract, you may remember this because you
remember it
came
well.
down to Pasadena, and we were going to, I was certain we were going to be able finally to identify the elusive U1 RNA in yeast. By adding a complementary oligonucleotide, we knew the 5' splice sites were conserved more or less by that time, and that you would add this oligonucleotide and look for some RNA in the banding pattern on the gel that would shift, and that would say that's the one that's U1.
Yeah, the problem was, however, that two of that particular snRNA in yeast is huge.
It's huge, yeah.
It's not a small RNA.
It's huge, and Manny
And
Arias
U2 is
discovered that,
even-
right?
even
bigger, right,
And
right. And so
most
we
of
never
it's
saw the
not
shift, it was up at the
not
top of the
required,
gel. Yeah,
Yeah, she had made quintuple mutants of small RNAs that she had sequenced and isolated, and none of them
Had a phenotype.
were lethal.
so they were all
So,
not involved in splicing, but in other nuclear processes, yeah, yeah. Do you guys yet know what all those extra sequences in U1 and U2 actually do?
And it's not for lack of trying.
I know, okay.
Well, going back a little bit, just after the discovery of introns, so in 1977, it was found that tRNAs have introns, and that we had been looking at tRNAs in yeast, but sequencing them, but we didn't find any with introns. But it turns out that there were introns, and once I saw that, then in an experiment that Anita Hopper did and reported at the Cold Spring Harbor meeting, was that a particular mutant that Lee Hartwell had isolated on RNA1 and on RNA2 accumulated these precursors, and I jumped in on it. Maybe it was bad for him. Anyway, that everything worked in that system. We could sequence the precursor tRNAs. We got it.
Which were built up when you had this mutation that was not removing them,
yes,
right? And then you could see the longer precursors, right?
And we turned the sequences, and we immediately got an extract that spliced them. And that's just went on and on until for another 15 years, and finally we knew everything about there was to know about tRNA splicing. Almost no one works on it anymore.
Yeah.
But it had nothing to do with messenger RNA
Well,
splicing.
but it's very elegant.
Yeah,
It's just a different enzymology and a different pathway.
It's also completely different. It just has a few enzymes. It's not this huge structure of the splices. So it wasn't lost on us that we needed to do splicing in vitro with the yeast system.
Because it had been so powerful with the tRNAs.
Yes, and I felt like sort of like Napoleon throwing his troops at Moscow. I had all these great postdocs, and nobody could get this to work. Okay, and meanwhile the other guys, Phil Sharp's lab, Tom Menion's
lab in mammalian cells.
Mammalian got their systems to work, and largely because we were using could only use whole yeast extracts, whereas they could use nuclear extracts. Which are already considerably purified.
Yeah. So how did Andy Newman figure it out?
He worked out a different kind of protection system that an oligonucleotide would span the spliced junction.
So it was the detection that he improved.
The protection reaction. And he could see splicing if it occurred at 0.1% or less of the...
Yeah.
Our other substrate was actin, which we had sequenced, and it had an intron. There it's a 5' amino terminus end of the protein. And that was a beautiful substrate. It continued to be a beautiful substrate in splicing, but we all we saw were fragments of it in the yeast extract. But once Andy got this thing to work, it worked immediately.
Great.
And then the genetics could be brought to bear, and you couldn't do this
in the mammalian system. So the yeast genetics were really important. And one thing I want to point out where I really owe Chris a debt of gratitude was early on. So we'd proposed, first published in 1979, 1980, that we thought that these SNRPs that were the targets of autoantibodies might be involved in splicing because we could see the base pairing between the U1 5' and 5' splice site consensus sequences. But there were a lot of doubters out there. And there was one classic sort of editorial that appeared in Science that said, the gloss is off the small RNAs, at least for now.
I hope I didn't write that article.
No, you didn't. A guy named Roger Lewin wrote that article. You are not even quoted in it. Our colleague Hugh Robertson was quoted in it, and Norm Pace. They didn't believe it. But we persevered, and then came the beautiful genetic experiments performed in your lab and in Alan Weiner's lab that actually showed that if you mutated a substrate, a splicing substrate at the critical sequences where there's base pairing with the snRNAs, that you could get some sort of a phenotype, a mutant phenotype or a null phenotype, that you could then suppress those mutations by engineering complementary mutation into U1 or U2 snRNA. And I think that was at the point at which I think people believed that SNRPs were really involved in splicing. And it's very funny because I'd actually had a postdoc in my lab who had wanted to do that experiment like 2 years before. And I had sort of poo-pooed it, which in retrospect I find very puzzling because I was such an enthusiast about the base pairing between the bacterial and phage messenger RNAs and the 3' end of 16 sRNA. And John
Dunn.
Dunn, yes,
and
had
Bill
gone on to
Studier,
do the genetic suppression experiment on the ribosome messenger RNA interaction, and it had worked. And why I should have been doubtful and discouraged as postdoc from pursuing that, and then Alan Weiner's lab down the hall from me, Alan had his, I think it was a graduate student, pursue it, and it worked as well. But that was a real watershed, was getting that to work, and I'm very appreciative. Thank you.
Well, and also it was controversial because the base pairing we showed first was between you two and the so-called tac-tac box,
Yeah.
which everyone had stoutly maintained was
A yeast sort of
an
specialty.
anomaly
Yeah,
in
yeah.
yeast. And so I remember so well the Cold Spring Harbor meeting where we reported this. And I remember that the mammalian people were offended. They said, well, what are we supposed to make of this? And in that paper, actually, we had shown that if you go through the literature and figure out which RNAs had been analyzed that had branch point sequences identified, that you got the same consensus. And so finally it had to be accepted that it was conserved.
Yeah. And your in vitro system then was immensely
then
powerful for analyzing all the mutations.
Then we could go very, very fast. And so this was about 1985 when we finally got the in vitro system. A friend of mine from phage genetics days was Ed Brody. He had come to work in my lab as a sabbatical visitor. And he did the experiment, which I think was being done at about the same time in mammalian extracts as well. You make radioactive precursor messenger RNA from in vitro to a T7 RNA polymerase, and you put it into an extract, and you run it to gradient. And we could show with mutants and so forth that the active complex was so sedimenting, probably between 30 and 60 S.
It was huge.
Huge, yes.
Much bigger than any of the SNRs.
We could show that if you don't do splicing, you don't make this complex. That is, with a mutant substrate, you don't make the complex. So we actually, we published this in Science, and we named the spliceosome. And so it was clear that you could go faster and better with the yeast system than you could with the mammalian system.
Yeah. I mean, I think it's such a beautiful story because it shows how things coming in from different angles, the autoantibodies that precipitated the SNRPs, the genetics, both in terms of showing the base pairing and being useful, and the in vitro systems, the in vitro biochemistry, all sort of converge together to pretty much the picture that we have now of the spliceosome. The details, of course, have been fantastic to see them now because of cryo-EM, where we can actually see that these base pairing interactions between the snRNAs that were predicted, there they are for everyone to see.
I didn't
And
think
all
I
the
could ever live to see that.
I didn't think I would either.
structure.
Yeah, it's just like I didn't think I would ever see the sequence of the human genome.
Yes,
I'm not
of
good
course,
at
that
predicting
would be-
things like that, but it's absolutely amazing what can now be
Well,
done.
yes.
So Joan, we've concentrated on all of your wonderful contribution to the small nuclear RNAs, and splicing, I wanted to return to a remark I made earlier, but not explicitly, which was in the early days of our experiments, not knowing any better, we just individually cloned all the RNAs that came down with the anti-TNG antibodies. And the terrible surprise was that none of them had phenotypes when we deleted them. And as John pointed out, we had a paper where we made a sextuple mutant, and it also had no additional phenotype. And so it was both a frustration that we hadn't found the right RNAs, but also this question of what were these RNAs? And we now know that most of those were SNO RNAs, but you have made so many important contributions to those RNAs and others. I thought you could.
So in the process of characterizing the autoantibodies that specifically recognized and immunoprecipitated the splicing RNA protein complexes, the SNRPs, we knew that there were a lot of other kinds of autoantibodies out there. And it was, pretty straightforward to just go through and try to look at all the different specificities and see what, if you p32 labeled cells, so you had the RNAs labeled and then did an immunoprecipitation with some other kind of autoantibody, what would you get? And one of the main ones that appeared early on was called antifibrillarin against a nucleolar protein. And that was the one that turned out to precipitate many of these SNO RNAs, small nucleolar RNAs, that we now know contribute to ribosome biogenesis, many of them being guide RNAs that actually direct the addition of various modifications into specific sites in ribosomal RNA. So we did a lot of that early on. As a matter of fact, we went from SNO RNAs to various other kinds of RNAs, and the question has always been, what does it do? What's its function? And it was a lot of fun exploring what many of the small nucleolar RNAs do with respect to ribosome biogenesis. And then, the wonderful thing about the RNA field is that for the entire time that I've been in this field, there's always something new. There's always a new RNA that is a non-coding RNA, most of them, and you don't know what it does. So there's a whole nother sphere to be experimented on and to make discoveries in about yet other things that RNAs can do. So even though my career has been very monolithic, and at this point I like to think about base pairing because that's the obvious thing that RNA can do, there are always new kinds of RNA and that need to be explored and that people need to figure out how they're contributing to cellular metabolism and regulation. So
but harder
it's been
experiments.
Lots and lots of fun, yeah. And there is, of course, also the second spliceosome, which is a story where one of the other people in that field, Rick Paget, in fact had published a paper, and I think maybe we were referees of the paper, where he had proposed that these minor low abundance SNRPs that clearly had all the same properties as the splicing SNRPs, that there might be a second kind of spliceosome, and I sort of poo-pooed that initially, but then later, with the right postdoc in the lab and doing the right experiments, it ended up that we worked on that, and for a while. So actually a student in my lab, just by looking at low abundance SNRPs that were precipitated by the same antibody, the SM antibody, as the splicing SNRPs, had discovered these low abundance ones. And luckily she named them U11 and U12 in the correct order, because U11 turned out to be the homologue of U1 and U12 turned out to be the homologue of U2. This was Karen Monska-Wasserman. And then we were actually quite surprised because we had thought initially, well, so maybe now you just identify the ends of the intron, with U11 and U12, and they have slightly deviant consensus sequences. And then U456 would be the same complex because we already knew that that was sort of the catalytic core of the spliceosome. And then it turned out that there was a different U4 and a different U6, but U5 is the one snRNA that's preserved in both spliceosomes. And now that we know the structure and we know all the things that U5 is doing at both steps in the splicing pathway, it's obvious why it might have been the one that was in fact preserved for the second spliceosome as well. So that was lots of fun.
Wonderful.
And then meanwhile, we were stumbling into looking at viral encoded, non-coding RNAs and trying to figure out what they might be doing for viral infection. And that's given rise to a lot of interesting things. I don't know that it's another field, but again, viruses are very inventive and they do things that you wouldn't expect host cells to do, but they always seem to, by figuring out what they do and how they do it, you cast light on processes in the host cell, and that's been a
So
wonderful.
going forward, is that an area you still want to focus on?
Yeah, there are a couple of problems there that I'm pretty excited about. I mean, one is one which is just sort of coming into focus now. When you think about microRNA populations, and we know almost nothing about how their degradation is controlled, it's quite clear that all the things they interact with, all the messenger RNAs and all the non-coding RNAs, have the potential for being the instruments of destruction and regulation of these microRNA populations. And that's something I'd like to know a lot more about, because there are interesting things going on there that we haven't yet delved into. Unexplored things. And we also stumbled into the area of RNA triple helices, which was a structural feature that was predicted to exist in RNA in 1957 there's a classic paper by Alex Rich, David Davies, and Gary Felsenfeld, but then nobody saw them in any naturally occurring RNA until we stumbled across it using a viral non-coding RNA in 2010. So, and I still want to know why there aren't more that we're finding and what they're doing.
So lots of excitement
Lots
ahead.
of excitement ahead, yeah. As far as I'm concerned, I'm always excited by something. So we want to say a word about women in science. The RNA field is much more populated by women scientists than many, many other fields. I don't know really why. Maybe you have a guess about that, John?
I could hazard a guess about that. It's something that you said right from the beginning, and that is that we didn't try to bring other people in the field down. It was always the opposite of trying to make them succeed. And so when we started the RNA Society, we had meetings in which we were there, and everybody stayed and listened to the talks. It's a wonderful thing. And we have a journal that's a respectable journal, and it's a fabulous success. And it made this field different.
I think you're right.
It has actually a favorable imprint, it seems to me, for women graduate students and postdoctoral fellows. And that could explain why there are more.
I agree, thank you, yeah.
Founder effect.
Yeah.
I just want to add an exclamation point to the potential role of women interacting with other women in support.
Yeah.
I went through a very difficult period early on in my career, and an outgrowth of that struggle was the formation of a group which still goes on to this day. Actually, Joan on sabbatical has often attended it, and it meets every other week, and it's going on for 30 plus years. And I think it's just been a remarkable source for all of us about how to succeed.
Yeah. So Chris knows, I know, that one encounters these awkward situations where people say things or do things that are inappropriate. And I guess my advice to other people struggling with the same problems is that what carries you through is your science and just doing good science and perhaps trying not to worry too much about what some of these negative influences are. That doesn't mean it's easy, and it's certainly very useful to have other women to talk to. Another thing that I think has been very special for us is the RNA field. And I must say, it's so different now from the way it was when we started, when you could count on the fingers of one hand the women that you knew that had regular faculty positions at any research university, and to now have women colleagues in your department. I have some, they're all much younger than I am, but it's still wonderful to have women colleagues in my department.
Yeah.
It just makes a whole lot of difference in very subtle ways that I find hard to enunciate.
Yeah, yeah.
It's just a feeling of belonging and that you're not something weird.
Right.
And thus to be discredited.
Right. Well, thanks, Joan, for sharing all your experiences and your wisdom.
Well, thank
been
you
really a
both
pleasure.
for being such a vital part of this, because I really hoped that we would get out the story of how things came together early in splicing, that now we understand this process, at least in its
Broad
outlines, right, as you say. And it's something I never thought, as John said earlier, I never thought would ever happen, that we would ever understand this complicated machine.
What I would like to say about this conversation that the three of us are having is that it's a conversation that's been going on now for 40 years, and I expect the conversation to continue as long as we have. But I have loved the conversation that we've had and the life we've shared.
Thank you both for being here.
Great.